Hi, my dear students. Here we're going to start discussing the topic of cell injury. This is a rather large topic, so you'll see that I have divided it up into three subtopics. They are hypoxia, cell adaptations, and then finally cell death. So the very first big message I'd like to get across to you as we dive into pathophysiology here is that disease is always going to occur at the level of the cell. So that is the basic unit of disease. So do not forget that throughout your time in your pathophysiology classes. And as you can imagine, how much injury occurs to a cell is going to depend on how intense or severe the injury is. And also, we're going to see which particular cell types are involved. So some cell types are more prone to suffering from cell injury, while others can deal with it much better. So what's going to happen is that a cell can tolerate injury up to a point, but then when the stressors, the stress causing things, when they get too overwhelming, we're going to see cell injury, which we can divide up into reversible and irreversible injury. And you'll see how capable of how capable cells are of actually tolerating injury and have the ability of somewhat reversing to their normal functioning. Of course, irreversible injury is going to imply cell death. And so this slide sort of reiterates what I just said about reversible and irreversible cell injury. Um, with regards to reversible cell injury, we're going to see how cells can actually adapt after they have been injured. They might go through various changes that we'll look at in that particular subtopic. And then within cell death, there are at least two major forms that we will study. They're very different in their physiology. Now, a cell can experience very direct injury or a more indirect type of injury, and you see the various examples of indirect injury, and we will not discuss all of those. We will discuss some of them later, as I indicate. In our discussion on cell injury, we will briefly touch on how the environment and also either overeating or not eating enough can create indirect injury. But we're first going to focus on direct injury, which is caused by a low oxygen environment for cells. And the term we use for that is hypoxia. So there's different ways in which cells can experience hypoxia, and that's what we're going to take a look at next. So real quick, before we take a look at how cells can experience hypoxia, there's different forms. Um, the question can be asked, cells that experience hypoxia, can they reverse to normal functioning? And yes, but it all depends on how long and the oxygen deficit goes on, how intense it is, how much damage it causes. And as mentioned earlier, some cell types are much more prone to cell injury. And if you think about it, for instance, cells that can't divide or cells that make up very vital organs, organs that have major functions regarding regulating such things as blood volume and blood pressure, they're going to be very sensitive. So the three major organs and therefore their cells that are going to be quite sensitive to hypoxia are heart cells, your brain cells, meaning neurons, and then so also your kidney cells. Your brain really cannot do very long without oxygen, maybe 4 to 6 minutes. Those neurons are very demanding in oxygen and they really cannot kick into anaerobic metabolism at all. And obviously, if the heart goes without oxygen for a bit too long, we might end up with a stroke or possibly a myocardial infarction. Um, and the problem is that, particularly with heart cells and brain cells, is that these are the kinds of cells that can't divide and can't replace themselves. So it creates a lot of damage in the organ. Now, there are some cells that replace themselves very easily, and they can also go much longer without ATP. And very good examples of those are your epithelial cells. They are tough little guys. Okay, so we're finally ready to really dive into the topic of hypoxia. And as I mentioned earlier, hypoxia is a form of direct cell injury, and it's the most common cause of cell injury. And so what happens when a cell experiences hypoxia? Well, it doesn't have enough oxygen or no oxygen, and obviously it cannot go through aerobic respiration. It cannot go through the process of the Krebs cycle with the electron transport chain to create ATP. And so we see that there's going to be a drop in ATP production, and that's going to have all kinds of consequences. So just as a quick reminder, in the process of cellular respiration, remember that we have two parts. We have the part called aerobic respiration, and that is going to require oxygen as its final electron acceptor to ultimately produce a bunch of ATP. And if there isn't enough oxygen or no oxygen present, then we can't kick into the Krebs cycle as well as the electron transfer chain, and we go into a form of fermentation. So to review this really quickly, whether oxygen is present or not, we're always going to see that we break down glucose in the form of glycolysis, right? And that's going to start out with a 6-carbon glucose, and we end up with a 3-carbon, or two of them, pyruvic acids molecules. Now, this is the time that if oxygen is present, and only then can we move on. And that is move on in the sense of going into the Krebs cycle. So if this here represents the inner membrane of a mitochondrion, because remember a mitochondrion has two membranes, then we see that this is going to allow for the location of our electron transport chain. Of course, that's going to happen after the Krebs cycle. So the pyruvic acid molecules are going to get prepped to enter into the Krebs cycle. And during this process, by the way, we also see some ATP produced. Don't forget that in glycolysis, we also have some ATP molecules produced. But it is really when we get to our electron transport chain, when oxygen can function as the electron acceptor, that we can crank out quite a bit of, where are we here? We can crank out quite a bit of ATP, right? With the hydrogen pumps, etc. We're not going to go in all of that detail again. So for all these three parts of aerobic respiration to be able to occur, we need oxygen. We'll talk about the lactic acid fermentation in another slide. So as I mentioned before, there are different ways in which cells and the tissues with the cells can experience a low supply of oxygen or experience hypoxia. So there's 4 different types, and on this slide we just have 2 listed. We'll list 2 more on the next slide. So the first one is referred to as hypoxemic hypoxia, and so you already know what hypoxia means. That refers to a low level of oxygen, but the first word tells you that we're dealing with blood. So emic or emia always means blood. I always like to give you the root of words to help you remember better what they all mean. So in this situation, we see that the tiny little blood vessels that we call capillaries, they're just microscopically very small and short, they have plenty of blood in them. So there's plenty of blood arriving. So there's very good perfusion, or there's good perfusion. That's what we mean by perfusion, good blood flow. But for some reason, there's low oxygen in the capillaries. And the cause of this low level of oxygen has to do with the lungs. So something is not functioning well in the lungs. And of course, the lungs are made up of little air sacs we call alveoli. So there's a problem there that prevents for enough oxygen to diffuse into the blood, and consequently we have something that we call hypoxemic hypoxia. The second type of hypoxia you're probably most familiar with. You may have heard it being called ischemia. Um, and this refers to the fact that there's literally poor flow of blood in the tissues. Maybe there's not a lot of blood arriving. And there's all kinds of reasons for why this might happen, which is not going to be our focus here. Um, and so that's going to create issues with oxygen levels in the tissues. We have two more types of hypoxia to discuss. We have a couple more ways in which tissues can suffer from hypoxia, and that has to do with the tissues themselves having low oxygen levels. So the third form is called anemic hypoxia. And again, this time we see that there's low levels of oxygen in the tissues, but the reason this is happening is because somehow your blood itself is struggling with carrying enough oxygen. And so this typically has to do with, for instance, um, oxygen not being able to bind to hemoglobin properly. You might have heard of people suffering from sickle cell anemia, and the name of the disease reflects the shape of the red blood cells. They're sickle shaped, and because of that, it's difficult for oxygen to bind properly to their hemoglobin. We will learn in much more detail about sickle cell anemia. We may also see that the hemoglobin molecules are all healthy and fine, but the spot on hemoglobin where oxygen is to normally bind is now taken up by a poison called carbon monoxide. You know, in the winter, there are always some people that die of carbon monoxide because they can't afford proper heating, and so they might turn on um, things in their homes that are not very safe, that produce all kinds of carbon monoxide. And then the carbon monoxide will literally compete with oxygen for hemoglobin. So that is called anemic hypoxia. Once again, you see that root there of emia, and the A means literally without blood. And that'll make more sense when we study the different types of anemia, why it's given that terminology. And then finally, we have something called histotoxic hypoxia. And you know that histo refers to histology, that is the tissue. So somehow there's some level of toxicity in the tissues such that they cannot um, use oxygen. So literally the oxygen cannot leave the bloodstream and make it in the tissue cells. So something is interfering with that mechanism of diffusion of oxygen from the blood, from the hemoglobin in the red blood cells into the tissues. And this is typically caused by a variety of poisons that might block your electron transport chain. You might have heard of cyanide. Cyanide is the poison that Hitler used to commit suicide. He also shot himself in addition to that, but this was a common method of suicide in the older days. So I'm going to say it again. Hypoxia is going to impact how well we can produce ATP by means of aerobic respiration. So ATP is a big, big, important molecule in our body. It has many critical functions. And so if we don't have enough ATP, we cannot do a lot of these functions. And so we have listed the consequences of hypoxic injury here, of course, primarily due to the fact that we don't have ATP. And so we're going to visit each one of these topics. So we're going to see that ATP is going to impact normal cell signaling. You know, our ion pumps, our sodium potassium pumps, for instance, they need ATP. They're going to struggle. Um, if we don't have a good enough level of oxygen, our cells are forced to kick into fermentation. So we're going to collect acid, namely lactic acid. And then we're going to discuss a couple of topics that might be less familiar to you. We'll briefly talk about something called heat shock proteins and the accumulation of reactive oxidant species, which contain free radicals, which can be quite damaging to cells. So we're now going to progressively go through these different effects of hypoxia that we just listed on that previous slide. And the first thing on the list was, of course, how ATP impacts cell signaling. And you might not have learned about this before, but ATP actually plays an important role too as a neurotransmitter. We tend to think mostly of ATP as a molecule that, when it splits by means of hydrolysis into ADP and its inorganic phosphate, it releases a bunch of cellular energy um, to make ion pumps function. Perhaps that's what you focused on primarily, but ATP is so much more than that. So it functions then as a neurotransmitter, for instance, and then it also plays a role in cell signaling um, by helping out enzymes such as kinases. So ATP can serve as a substrate for these kinases so that they can then phosphorylate other substrates. And this sets off a whole cascade of events. And again, you'll see this as we continue to study things in pathophysiology. This cascade of events is then going to lead to a cell starting a major metabolic function or perhaps inhibiting a major metabolic function. Here I list as an example protein synthesis. Another type of substrate that that enzymes use, another enzyme that uses ATP as a substrate is called adenyl cyclase. Sometimes you see it spelled slightly different, which plays a very important role in something called G protein signaling, which you were introduced to in the cell review. So if this is a little foreign to you, the G protein signaling, you need to go back to the cell review slides that I provided to you, because we will talk about G proteins quite a bit in this class. So in addition to cell signaling, ATP plays a very important role in keeping ion pumps going. You know this by now. But one of the things you might be as familiar with is that we don't just have sodium-potassium pumps, but we have calcium pumps, we have hydrogen pumps. You've seen the hydrogen or the proton pumps already. We have chloride pumps. So there are lots of ion pumps in the cell membranes of all cells. You know, these ion pumps are always going, they're always pumping, and they play a very important role in maintaining a cell's resting membrane potential. Remember that all cells have a resting membrane potential, all of them. But it's only excitable cells that can undergo a change in their resting membrane potential. Those are your muscle cells, and those are your nerve cells, also called neurons. These ion pumps play a very important role in maintaining the resting membrane potential by pumping their ions against their concentration gradients. I don't think we need to go over that. You've learned about this before. One of the things you do need to be aware of and take with you for the rest of your career in healthcare is that these ion pumps, they suck up most of a cell's ATP. So what I'm saying here is that all that ATP that we produce via aerobic respiration, most of that is used by these ion pumps. And so if those ion pumps can't function, they cannot regulate all of our cells' resting membrane potentials. And now we're going to get in serious trouble. We're going to see, especially in the case of our discussion on electrolyte imbalances, how important it is in order for ion concentrations across the cell membrane to be just right. So in your cell review slides, you really, or it was emphasized that you really had to memorize the concentration gradients of 4 major ions, and they're listed here. When it comes to the sodium-potassium pumps, when they fail, it's really going to impact the sodium levels, much more so than the potassium levels. Because of how many sodium ions are pumped across the cell membrane. That is 3 to 2 for potassium. So what's happening without the proper levels of ATP is that after sodium has leaked into a cell, via, let me use a pen here, when it leaks into the cell via just the leakage channels, um, that sodium cannot stay there. It needs to be pushed back out, right? Let's say that here we have a pump, our sodium potassium pump. Then that sodium needs to be pushed out against its concentration gradient with the help of ATP, so that we can keep all of these gradients, meaning with too much or more sodium outside than the inside. So if we don't have this ATP, this can't happen, and we begin to accumulate more and more and more sodium on the inside. So let's do that. So we have too much sodium on the inside now. It gets literally crowded with sodium ions. And water will always want to go where uh, our sodium ions are. Water likes to follow salt. Let's put it that way. Just put that in a little area in your brain and don't ever forget it. Water loves to follow salt. Water loves to follow sodium, therefore. And so we're going to see that happening, that water will start to follow by means of osmosis. Remember, water always moves by osmosis, and it's going to go towards where there's a high solute concentration. And of course, that's going to cause for the cell to swell, and we will refer to this process as hydropic swelling. This is an actual phenomenon that occurs in cells due to too much influx of sodium ions. And we'll repeat all of this again, this process of hydropic swelling, when we focus on body fluid imbalances a little bit further down the road. This has all kinds of consequences, as you can imagine. So here we go. Let's take a look at the consequences of the cell starting to swell because all this water is following the sodium. Well, it's going to cause for the rough endoplasmic reticulum to start dilating, and that causes for the ribosomes that are attached to the rough endoplasmic reticulum. I guess we could just add that here, the R for rough endoplasmic reticulum. Because literally your rough endoplasmic reticulum is just also filling up. And now we don't have the machinery to go through the process of protein synthesis. So now we have a problem with protein synthesis. If we have a problem with protein synthesis, we can't produce or synthesize all kinds of proteins, including enzymes. And especially, we're going to see that this affects the cell membrane, because what is the cell membrane made up of? Remember that your cell membrane is made up of two major molecules, and those are proteins, I'm abbreviating, and also phospholipids, right? Again, I'm abbreviating. Don't forget that all of these basic biology topics that you learned about, you're going to have to bring in to your pathophysiology class here. So the cell cannot repair cell membranes. And what are these proteins in the cell membrane? A lot of those are receptors for all kinds of hormones or neurotransmitters, but also all of your ion channels are proteins. And I'm not talking just ion pumps. I'm talking about all these channels you've learned about in AMP1, such as your chemically gated channels, mechanically gated channels, your voltage gated channels, your leakage channels. Those are all protein based. And so now we don't have all of these ion channels controlling what enters and leaves the cell. And we're going to see that there's major leakage in the cell membrane. I'm abbreviating, I think you know by now, cell membrane by a CM. Most often I use small letters, but so the cell membrane leaks. We're going to see that all of the ion concentration gradients, the water gradients are all messed up now. And this ultimately leads to another problem, that is mitochondrial swelling. So we're going to focus some more on this problem, and that is mitochondrial swelling on the next slide. But let's stop here for a moment. Remember that we started this this discussion here today by stating that one, all disease occurs at the cell level, and two, that there are two forms of damage or injury to cells, right? We can see reversible injury or irreversible injury. So we've reached a point of the cell still being able to reverse its injuries and go back to functioning somewhat normal. So if oxygen levels return to normal and the cell can start cranking out enough ATP, it can fix itself more or less and actually deal with some of the damage that's been done to its membranes. Meaning not just the cell membrane, but even the endoplasmic reticulum, the mitochondria can recover, etc. But if, on the other hand, hypoxia continues, it's not going to get so pretty. So if hypoxia continues, there will be further damage to the cell membrane. It becomes even more permeable. And we're going to see something called calcium overload. So you're going to see this term a lot in the future. The moment you see calcium overload, you know from now on, that's my dog shaking, that the cell is in trouble. It is now being threatened of dying. So to repeat here, when we see continual hypoxia, then we're going to see that the additional ion pumps, namely calcium ion pumps, are going to also fail. Of course, they have begun to fail along with the sodium potassium pumps, but um, those sodium potassium pumps are very critical to maintaining resting membrane potentials, remember. But when those calcium ion pumps on top of that begin to fail as well, making the cell membrane even more permeable, particularly to calcium, all of this calcium, remember the calcium gradient? When you look at a cell, we're going to have not only much more sodium, concentration of sodium is much higher on the outside than the inside, but it's the similar thing for calcium. And remember, calcium has two positive charges. So much more calcium on the outside of a cell than the inside of a cell. So by passive transport, both of these ions easily leak into the cell, relatively easily. Not as easily. Potassium of all these ions moves across the cell membrane the easiest. Of course, potassium moves out. But because of its concentration gradient and because of the cell membrane having become so leaky and because of the calcium ion pumps failing now, we're seeing that there's way too much calcium accumulating inside of cells. And this is going to really, really damage mitochondria. And it's going to activate very destructive enzymes. And we're now looking at the death of the cell. So with all of this excess calcium inside of the cytoplasm, we see that that actually triggers the creation of these little pores on the inner membrane of mitochondria. So these little pores form, and that is going to allow for all kinds of things to leak into the mitochondria, including a ton of calcium. Your mitochondria swell, and all kinds of consequences occur, such that not only will your calcium ions leak their calcium into the cytoplasm, we're going to see that because many of the uh, substances that make up the electron transport chain are going to participate in the formation of ROS, and we will see what exactly that is in just a few minutes. They have something to do with free radicals. And of course, now that our mitochondria are damaged, we're going to have even more problems with ATP production. And that is going to typically lead to a form of cell death we call apoptosis. There are two forms of cell death, but when the mitochondria are damaged by a calcium overload, it's typically going to lead to apoptosis. So keep that in mind. So what are these destructive enzymes that are released in response to the calcium overload? Well, we're going to see that these protein lysing enzymes are produced, better called proteases, so they break down proteins. And you know, anything that breaks down proteins is going to have major consequences on the functioning of the cell, but particularly what's going to be impacted almost immediately is the cytoskeleton of the cell. So the cytoskeleton of the cell starts to fall apart. Imagine that our skeleton starts to deteriorate. We could not function anymore either. In addition, phospholipase enzymes, and as I said earlier, your cell membrane is made up of proteins, which are also attacked by these proteases, not just the cytoskeleton. And the second major type of molecule in a cell membrane is a phospholipid. So now those are falling apart or being destroyed. And that's, by the way, when these phospholipids um, are broken down, we're going to see that proteins from the injured cells are now leaking into the blood. And so when we start to see proteins leaking into the blood, that typically will trigger another form or an additional form of cell death called necrosis. And we can look for these proteins in the serum of a patient and predict that more than likely some level of necrosis is occurring in the body of that patient. So that's why we say that they are useful, form a useful marker. Oh boy, a flow chart that looks overwhelming. Well, it's not going to be once we've walked you through it. And so far, we haven't studied all of the parts of this flow chart, so we'll keep coming back to it. So grab a pencil. Hopefully, you have a way to mark up this flow chart on a piece of paper or on your device that you can use a stylus with, because that's what you'll need to do to be able to better understand this flow chart. So let's go through it. So here we start with the problem, hypoxic injury. We see a decrease in aerobic metabolism that, of course, leads to a decrease in ATP synthesis, which then creates all of these issues that we're seeing on your screen right now. I've not listed every single detail, obviously, but remember that if we don't have enough ATP, we're going to have major problems with cell signaling. So you see that here. And then so far, we have especially focused on the problem with a decrease in active transport, meaning those ion pumps can't function anymore, right? When it comes to your sodium potassium pumps, they're going to cause for sodium to collect in cells and water follows, and now we see hydropic swelling. When we see hydropic swelling, that is going to ultimately lead to problems with your ref endoplasmic reticulum dilating, your ribosomes detach, and poof, we have now problems with protein synthesis. In addition to that, um, we're going to see that little by little mitochondria begin to swell. And at first, that's not going to create a huge problem. But if hypoxia continues, remember, we're going to see that the calcium voltage gated channels begin to fail. So even more calcium accumulates, leading to more mitochondrial swelling. And ultimately, we're going to see problems with the activation of enzymes that destroy the cytoskeleton as well as the phospholipids that are making up the cell membrane, and that leads to cell death. It could be apoptosis, necrosis, it could be a combination of the two. So we're now going to take a closer look at these other parts that are consequences of hypoxia. So it's pretty obvious if a cell doesn't have enough oxygen, it cannot easily go into aerobic respiration to make ATP. So some cells who have that ability will switch over to lactic acid fermentation, because even without the presence of oxygen, a cell can kick into glycolysis, right, which still produces some ATP. the second part of that is lactic acid fermentation. Keep in mind that does not produce any ATP. Only glycolysis does. At any rate, so now we have the buildup of lactic acid, and of course, acid is going to mess around with the pH. The pH, an acidic pH is going to be a pH of less than 7. So the intracellular pH is going to drop, and we all know by now that proteins, enzymes are proteins, hate for the most part a low acidic pH unless we're in the stomach, let's say. It's also going to have another consequence, and that is that lysosomes this time are going to swell, and they're going to pop, and they're going to leak their enzymes. And of course, lysozymes contain enzymes that are very destructive. And now we're going to literally see that the cell begins to digest itself, and that is called autophagy. Auto meaning self, phagy, eating, self-eating, right? Self-eating. You've heard of cells that are called macrophages. They're big eating cells. So quick reminder again, if a cell has access to glucose, which is a 6-carbon molecule, it can easily break it down into two pyruvic acid molecules that are 3-carbon molecules, as long as there's a bit of ATP present, and then the net production is 2 ATPs per glucose. So we need to have some ATP to get this going. Do we need oxygen? We don't, right? So glycolysis doesn't care whether oxygen is present or no oxygen is present. Keep that in mind. If oxygen is present, we can kick into aerobic respiration with the Krebs cycle and the electron transport chain. Krebs cycle here, also called the citric acid cycle, remember, and then our electron transport chain here. That cranks out a lot of ATP. However, often a cell does not have enough access to oxygen, or we are here discussing hypoxia. And so cells that can do this are going to now have to depend, or I should say they're forced to kick into anaerobic respiration. And so this is the situation where there's not enough or no oxygen present. And in our body, we're going to see that this creates a buildup of lactic acid, right? We're not looking at alcohol fermentation, even though we have bacteria and yeast that could do that in our body. But when it comes to our own cells, it's going to be lactic acid fermentation. Keep in mind that fermentation does not produce any ATP. So the only time or the only place where some ATP is formed if there is not enough oxygen is during glycolysis. And as you can imagine, that is used up really fast. So a cell runs out very quickly of ATP if it has to depend on just glycolysis. So now we're seeing the buildup of all of this lactic acid, and of course that is going to decrease the pH of the cell. And so we're back to our flow chart. And just a moment ago, we covered all of this side. Where am I here? Sorry. We covered all of this side of our flow chart. But now we're going to focus on this portion. So if there's not enough ATP, the cell will go into anaerobic metabolism, and consequently the pH drops, and we're going to see problems with the lysosomes really cannot hold on to their membrane, so all the contents, all their enzymes leak out, and those are typically proteolytic enzymes, which are of course going to break down all kinds of things in the cell again, such as the cell loses its cytoskeleton integrity, and we see cell death again. Hypoxia does have something else interesting happening, and that is the activation of so-called heat shock proteins. And they guess that name because they were first discovered as being proteins activated in response to heat. But we now also know that they become activated in response to hypoxic injury. You can think of these as proteins that are chaperone proteins for so they themselves are proteins for other proteins. So they do all kinds of great things in making sure that proteins fold correctly. They make sure that proteins that were damaged will be folded up again properly, right? So they have the ability to do some repairing in response to hypoxic injury. So that leads us then to the final effect that we had on our list regarding the damaging effects of hypoxia. And that is that hypoxia causes something called oxidative stress, or you may see it being referred to as respiratory burst. So try to be very familiar with these two terms. And what are they all about? It refers to the release of ROS, and you've heard me mention it earlier, which stands for reactive oxygen species. So we're going to study these in a little bit more detail and what effects they have. So when a cell is beginning to accumulate too many of these ROS, these reactive oxygen species, it pretty quickly gets in trouble. Because it means that what we call antioxidants are not successful in removing the ROS at a fast enough speed, and the cell cannot recuperate from the damage that the ROS are doing. Now, these reactive oxygen species are not all bad or not always bad. They're actually normal molecules present in our cells, but it's important that they are always kept at a low level, and that they are, when they are produced and secreted, that they're rapidly removed. And that's why we have what we call antioxidants. And that's why, you're often informed, oh, you should eat a lot of, thinking of something, blueberries, because they contain a lot of antioxidants. So now you're going to see their importance. So what are these ROS, or reactive oxygen species? Well, for one, they all contain oxygen in some form or another, and they have the ability to steal electrons. So many of them are literally free radicals. And so here you see a list of examples of reactive oxygen species. And perhaps you'll be surprised to see some of these. For instance, hydrogen peroxide, that we may still be using, probably shouldn't so much, to treat minor wounds, is a type of an ROS or ROS. Remember that when you took introductory biology, you used hydrogen peroxide with liver juice, right? Because liver juice contains an enzyme called catalase that rapidly breaks down hydrogen peroxide into two harmless molecules, water and oxygen, and also produces heat. And it's the heat that we depend on to kill bacteria when we have a little cut that we put hydrogen peroxide on, by the way. So we have this enzyme in pretty much all of our cells to rapidly break down hydrogen peroxide, because if it does accumulate, it can create havoc, and we'll look at that. You might have also heard of nitric oxide, which we abbreviate as NO. And in our body, it plays a very important role, for instance, in vasodilation. It's also an important chemical signaling molecule. If you are in AMP2, you'll learn about the importance of nitric oxide and vasodilation. And then there's a bunch of other ones, hydroxide ions, you've probably heard about too. So I do need for you to know all of these examples. I'm not really needing for you to know the detailed structures that are illustrated here, but not um, it's not going to hurt you to better understand their structures. So as I tried to explain to you, these ROS molecules are not all bad. As a matter of fact, they're normal byproducts of cellular respiration uh, in mitochondria and other organelles called peroxisomes. They're always present, as I said, at a low and stable level in healthy cells because we have like catalase enzymes that can rapidly destroy them. We depend on ROS as signaling factors and protective factors. For instance, with regards to the immune system, you'll see that ROS are secreted by immune cells, or phagocytic cells, I should say, to help with digesting pathogens, for instance. And then, as I already mentioned, nitric oxide, this is an abbreviation for nitric oxide. So this is not NO, but nitric oxide. It's an important vasodilator and also a signaler, a chemical signaler. But if ROS do accumulate in cells, if the cells do not have the ability to remove ROS fast enough, unfortunately, they can do some serious damage, and they're going to go after all four of our biomolecules. I'm not listing the impact on all four of them, but I impact at least your nucleic acid, so it's going to start messing around with DNA to where it can be mutated. It can start changing how proteins are folded or break them down. And again, ROS can impact lipids so that they go through something called lipid peroxidation, which creates, again, chemicals that can steal electrons from lipids in cell membranes. So let's come back to our flow chart because we are done with it now. What we covered in the last few minutes here is how hypoxia will ultimately activate heat shock proteins. And hypoxia can also lead to the accumulation of these ROS molecules, these reactive oxygen species, which can, when they do accumulate in large amounts, can literally destroy all of our biomolecules, I should say. Whether it's our DNA, nucleic acids, that is, whether it is the lipids, the proteins, even carbohydrates. So we have finished this whole flowchart. So here we have cells producing ROS, which are damaging. Why are cells doing that? Well, it is a defense mechanism to accumulate, or I should say, to not to accumulate, to have a way of producing substances that can destroy pathogens or can get rid of cells that are damaged and or old. So we need these chemicals called ROS, but to keep them under control, we also have evolved other chemicals that are called antioxidants. So their focus, their goal is to literally inhibit or neutralize ROS. And so we've already mentioned some of them. Enzymes such as catalase and peroxidase can break up some of these ROS. We have various proteins that can bind with metal ions, such as iron, and these metal ions are really good at acting as free radicals. So they can steal electrons and set up this chain reaction that can destroy cells. So by keeping these metal ions bound to proteins, they cannot do that as easily, if at all. Various vitamins, A, E, and C, function as antioxidants. We have some elements, selenium and zinc, that are antioxidants. And then even some chemicals in plants. You might have heard of flavonoids, especially, maybe not as much of glutathione, but especially flavonoids are healthy antioxidants that we obtain from eating veggies. So this wraps up our discussion of the direct consequences of hypoxia. In the next video, we're going to focus more on the causes of oxidative stress, for which we need to list hypoxia, obviously, as well.